Yeah, hi, hello, welcome to our YouTube channel. Myself Sindhil Kumar, biology teacher. Today we are going to deal about the tools of recombinant DNA technology from chapter Biotechnology Principle and Processes. Okay, this is the second video, the continuation of the first video. Okay, so here what are the tools we are going to utilize in a recombinant DNA technology? Okay, that is what the thing we are going to discuss in detail in this particular video. So, especially the restriction endonuclease or restriction enzymes. Okay, this is the enzyme which play a very important role in especially recombinant DNA technology. The another name of the restriction enzyme is molecular scissor. Okay, what is its role? It used to break the DNA fragment. Okay, break the entire sequence. If you take any DNA like this, it will cut the DNA into small fragment. That is the role of restriction enzyme. Okay, as per our desire. As per our desire, it will cut the DNA into small fragment. Okay, next, the first one, the next tool is going to be a cloning vector. Okay, so the cloning vector, by the word itself you could see, the very good example is PBR 322. It has 4363 base pair. Okay, it has a 4363 base pairs, and here this particular plasmid we are going to utilize as a vector to carry our gene of interest. Okay, whatever our desired gene we are going to insert in this particular vector. Okay, so this is one of the very good example. And what are the BAM-H1, SAL1, PVU1 and PST1? These are all the restriction enzymes. RE, just I will put RE. These are all the restriction enzyme. Okay, and this is a site for this enzyme. This is a site. This is a recognition site. This is the recognition site for those enzymes on the. Okay, it can bind this particular region and cut. It will cut this particular region. There, once it has been cut, in that particular place we are going to insert our gene of interest. Okay, one by one we are going to do. Next, third one is a competent host. What is the competent host? Means the host which can able to intake a foreign gene or our gene of interest, which can able to intake a foreign gene, that is called competent host. Okay, the competent host, to make a competent host we are supposed to follow various kind of protocol. Little later we are going to discuss about that. Okay, and next, here restriction enzyme, about the restriction enzyme in detail we are going to deal. What is restriction enzyme actually? The enzyme which cut DNA at specific site into fragments. Okay, so if you take that already we said no, we discussed. So if we take some sequence of DNA from human, we need of particular part of DNA. Okay, particular part means that is a gene which can express the particular protein. Okay, protein in the sense it can be any kind of protein, or maybe the insulin. Okay, can be insulin, or maybe a melanin pigmentation, melanin pigment. Like that, the gene have a capability to express a particular protein. Okay, so that is what the gene we are going to take for what is that? Recombinant DNA technology. Okay, so here the restriction enzyme which have a capability to cut at the particular end of the complete sequence. Okay, so they belong to, especially the restriction enzyme is that belong to the class of the enzyme called nuclease. Okay, and we have discussed what is restriction enzyme and how it works. Now we are going to deal when and how they have discovered the restriction enzymes. Okay, so the very first, in the year of 1963, the two enzymes responsible for restricting the growth of bacteriophage. Okay, what is bacteriophage actually? It is a kind of virus which can able to cause disease to bacteria. Okay, the E. coli is the bacteria, right? Here the coli is there. Okay, so the E. coli is the bacteria that can be affected by a bacteriophage. That is what? The bacteriophage is the virus which can able to cause a disease to the bacteria. Okay, so here especially the bacteriophage have a certain characteristic feature. What is that means? It doesn't have any cell organelle. That's why it is called an obligatory parasite. Okay, it completely depends on others' metabolism. It is completely depends on others' metabolism. Okay, so once it has been inserted into the E. coli, if just consider this is E. coli, main DNA and a plasmid DNA. If the bacteriophage, okay, just I'll draw the rough diagram. If the bacteriophage settled or pinged, okay, or used to be stick over the surface of this particular E. coli, then it release its genetic material. It's nothing but nucleic acid. The nucleic acid have a capable to enter inside, and what it does means it used to incorporate with the genetic material of the bacteria and it replicates. Okay, so let us deal with the HIV. The HIV virus we know, right? What its role actually? The HIV virus which has a RNA as a genetic material, which has a RNA as a genetic material. It contains a RNA. Okay, it have a capability to release the special kind of enzyme called reverse transcriptase. What is that? Reverse transcriptase enzyme. Okay, so what is the role of reverse transcriptase means? So the RNA is there, right? The RNA will be converted back into DNA. That is the role. Okay, so usually what happens in a central dogma of life, we discussed, right? DNA to mRNA. What happens? This process is called transcription. This process is called transcription. Okay, this is I. But because of this particular enzyme, what happens? The RNA will be converted into DNA, and once that HIV has been inserted into our body cell, with the help of the reverse transcriptase, the RNA will be converted back into DNA. That DNA of the virus will incorporated with our genetic material, and along with our genetic material it will replicate. This is how it used to replicate. Okay, so in similar way here also the bacteriophage is there, right? That genetic material will inserted into the bacteria and it replicates. Okay, if replicate in the sense it will not allow the further process of E. coli and it may kill that. Okay, it is toxic to that particular bacteria. Okay, to avoid that, what it does, especially E. coli does, it releases the two kind of enzyme. One enzyme, the one enzyme is a methyl group. Okay, it added the methyl group. What it does? It added the methyl group CH3, right? Okay, it added the methyl group on the surface of this particular genetic material. Okay, and the another enzyme is nothing but restriction endonuclease. Okay, so naturally it is exist in bacteria. Okay, that particular restriction endonuclease, what it does, it cut the DNA of a virus. Okay, it cut the DNA of a virus into small fragment. Because of that, the viral genetic material cannot able to incorporate. First thing, it has its own genetic material, right? The bacteria has its own genetic material. Above that, it synthesizes the methyl group. It gives a protection. Okay, the another enzyme is restriction endonuclease, which act on the viral DNA and it will be cut into small fragment. This is how it got a what is a resistant or protection from a invading microorganism or invading disease causing agent called bacteriophage. So what we have done, so we have isolated this particular restriction endonuclease very first in the year of 1963. This is what the enzyme. Okay. So in that way, more than 900 restriction enzymes have been isolated from over 230 strains of bacteria. That is the beauty. Okay. So the very first isolated from a E. coli. Okay. It has a resistant, right? Two kind of resistant. One adding a methyl group, and another thing is it releases the enzyme called restriction endonuclease, which act on a foreign DNA. Who is the foreign DNA? Bacterial DNA. Okay. It's act on a bacterial DNA and it will cut into small fragment. So then it cannot able to cause any disease to its own cell. Okay. Own cell means we are talking about bacterial cell. Okay. And say in the same way we have isolated the more than 900 restriction enzymes from a 230 strains. This is how we were isolated the restriction enzymes. Okay. And next, now we are going to deal how we have given a name. Now we are isolated. Once you are isolated any enzymes, we are supposed to give a name. Okay, in +1 you would have studied about the nomenclature methods and all, basic principle of nomenclature. Okay, we are supposed to follow some rules and regulation, right, some protocol of basic rules. So similarly here also, once it has been isolated, we are supposed to give a name. So then it can be universally accepted. Okay, so let us see here example E. cor 1. Okay, the E is nothing but a genus name, and CO coli is nothing but a species, and R is the strain, and this is nothing but a Roman letter. The one is a Roman letter. Okay. So the Roman number, why it has been given means the order in which the enzymes were isolated from the strain of the bacteria. Okay. In one bacteria we can able to get more than 2 strains also. Okay. So more than 2 enzymes also, not a strain, enzymes. Okay. We can get more than 2 enzymes also. Okay. In that case, we supposed to mention the Roman letter 1 enzyme, Roman letter 2 enzyme. For that purpose only, we have mentioned the form of Roman letter. Okay. Let us deal. The first letter indicates, again we will go with the same content. The first letter indicates the genus, and the second letter, 2 letter indicates the species, and third means in the sense next to that is represent the strain. Okay, and finally it represents the Roman letter. This particular E. cor 1 comes from the E. coli RY30. Okay, like that in E. coli itself the various types of E. colis are there. Okay, in that RY13 is the one among that. From that we have isolated the E. cor 1. Okay, this is a very familiarly using restriction enzymes in a recombinant DNA technology. And next see. So now we are going to deal about the types of restriction enzymes. There are 2 types of restriction enzyme is there. One exonuclease, another one is a endonuclease. What is exonuclease actually? They remove nucleotide from the end of the DNA. Okay, this is the DNA sequence. Okay, from the end it's cleave the DNA from the end to end. Okay, for example, if you take this is a 3' end and 5' end, this is going to be a 5', the another end is going to be a 3'. Okay, so this is actually the continuation of the double stranded DNA. For our understanding, I have made such a what is a base pairs for understanding how the restriction enzymes act on that. Okay, for that purpose the diagrammatic stands has been added here. Okay, so in that, especially the exonuclease, what it does, it act on a what is that end and it cleave that. But the endonuclease is contrast to that. What it does, it cut the specific position within DNA. Okay, so what it does, it bind to the specific recognition site of a DNA and cut the 2 strand at a specific point. That is the beauty. It cut both the strand. Okay, the DNA is a double strand, right? Okay, both the strand together it will cut. Okay. and see. So, now the continuation. The very first endonuclease is Hind2. This is the very first restriction endonuclease. Okay, and it cut the DNA by recognizing the specific sequence of 6 base pair about its characteristic feature. About the Hind2 characteristic feature, it can able to cut the 6 base pair. Okay, it recognizes the 6 base pair and it will cut that from a particular sequence of DNA. Okay, and this is called a recognition sequence for a Hind. This is what the thing, GT, PY, A, C. So better instead of this you can go with this. Okay, so it may be anything, G, T, A, C, C. Okay, here 6 is there. Yeah. So like that, any bases it can recognize and it will cut that. Like that, the various kind of enzymes are there. ALU1, HA3, BAM, H1, Hind3, ECOR1. Okay, so among that, the ALU1 and the HA3 is produce the blunt end. Okay, but the BAM H1, Hind3 and ECOR produce the streaky end. So depend upon the restriction in enzymes, its functions also differs. Okay, so some few restriction enzymes can produce a blunt end and some restriction enzymes can able to produce a streaky end. About the blunt end and streaky end little later we are going to discuss. Okay, and see. So this is a continuation. The restriction endonuclease recognize the specific palindromic nucleotide sequence. What is a palindromic? Especially you know about the MALAYA Malayalam. Okay, so if you read from a left side to right side or a right side to left side, its meaning is going to be similar, Malayalam. Okay, similarly in but we cannot say similar, but in a palindromic nucleotides, either it may be from a 5' to 3' direction or a 3' to 5' direction. Okay, see here GAA TTC in a single strand 5' to 3' direction, and in opposite also 5' to 3' direction you could see the say GAA TTC. Okay, it's both are similar, right? That is called a palindromic nucleotide. So it is a sequence of a base pair, that read the same on 2 strand. The sequence of both the strand is going to be similar. That's why it is called palindromic sequence. It may be a 5' to 3' and a 3' to 5' direction. Okay, so both are going to be similar. That is called palindromic sequence. Okay, so the restriction enzyme have a capability to recognize the palindromic sequence of both the single strand. Okay, and these are all the characteristic feature of restriction endonuclease we are discussing. And already we have discussed that we are going to deal about the streaky end. Now let us see the restriction enzymes cut the strand little away from the center of the palindromic site. Okay, see here, this is what the sequence. Okay, and it recognizing the particular sequence and it is not cutting at the center. If cut in this region, it may be considered as a center, but instead of that, it recognizing the particular base between the G and C in a just consider 5' and a 3', and similarly here 3' to 5'. The enzyme just come and recognize the particular sequence and cut the between the same base pair. G and A means here also is going to be a G and A. Okay, so but between the same 2 bases on the opposite strand, and this leave the single strand overhanging stretches at the end, and they are called as streaky end. Okay, so once it has been cut that particular double strand, you could identify the streaky end. See, it is hanging, right? So this hanging end, it is used to be hanger, so then easily it can able to bind with the another complementary base pair. Okay, for that purpose the streaky end is there. So that already we have discussed is depend upon the restriction enzymes. Okay, so some restriction enzymes may produce a blunt end. What is blunt end means? it will cut at the center and you won't get sticky end like this. Okay, if you have a DNA, if you cut in this particular position, okay, GATCC for GCTAGG, okay. If you cut at the center, okay, finally you will get only a GATCC and CTAGG. Then it is going to be, for example, it is going to be a end like this. Okay, so you are going to have only the blunt end, but sticky end means it seems like this. It is going to be a sticky. It is hanging, right? It is hanging in this particular double strand. This is called a sticky end, but this is called a blunt end. Okay, this is the difference between the sticky and a blunt end. But some the sticky end, especially why we are going for a sticky end means from a H bond, especially this stickiness facilitates the action of DNA ligase. Anyhow, in this particular end you are going to have a, what is that particular bases, with the bases the hydrogen bond used to be there, okay, that is between the two base pairs. These are all we are aware about that, but this particular sticky end favor the recognition site for the ligase enzyme. That is the beauty. Okay, so it facilitates the action of ligase, DNA ligase enzyme. When cut by the same restriction enzymes, the DNA fragment have the same kind of sticky end. They are joined by the ligase enzyme. That is the thing. What happens means, in this diagram we are going to discuss, if you take any foreign DNA and you have taken the vector also, vector DNA of the E. coli, and both the foreign DNA as well as the vector DNA, we are subjecting to the restriction endonuclease. It is supposed to be similar. The same restriction enzyme supposed to be used for a foreign DNA as well as the vector DNA. Once it has been used, what it does, it will cut into small fragment. Okay, so what happens, because of the function of restriction endonuclease, this foreign DNA has been fragmented into 3 pieces. In that, this particular region is our gene of interest. Gene of interest means our desired DNA. For example, it may synthesize the insulin. From the beginning, we are talking about insulin. Okay, so this synthesizes the insulin. Okay, now this is what the desired gene. That means our gene of interest to be inserted into plasmid vector. Okay, because of using of same restriction endonuclease, if suppose the for a insulin GAATTC, this is the, for example I am saying, this is the sequence which is responsible for a expression of insulin. Okay, so the restriction enzyme also designed in such a way. So restriction enzyme also recognize this particular sequence and cut that. This is what the scenario. And because of using the same restriction enzyme, the same kind of base pair it will identify and cut. So then we can able to insert our gene of interest into the plasmid. Okay, so once it has been subjected, now we are introducing our gene of interest into the plasmid vector, then it is called a recombinant DNA. It is nothing but rDNA. This is considered as rDNA. Okay, so after that what we do, the recombinant DNA is inserted back into the E. coli, into the host for a multiplication. It is nothing but cloning. So then we can able to get a more number of our gene of interest. This particular gene of interest for insulin, right, that we supposed to get in some thousands or lakhs or crore, whatever may be. Okay, for that purpose we are inserting back into the E. coli, and this particular E. coli is transferred into the culture medium. It may be in a liquid medium or a solid medium. Okay, once this has been inserted into the any culture medium. It may have a nutrition broth. Okay, this may have a proper nutrition. In that, if it has been inserted, then it will start at multiple. Multiple in the sense asexual reproduction will take place. The E. coli can able to produce its progeny within 20 minutes. Hardly it will take a 20 minutes to produce one progeny. Okay, see, it have a capable to produce 2 daughter cell, right? Okay, it will take only 20 minutes. That is the beauty of E. coli. So within a 20 minutes, our gene of interest also replicate. So initially we have inserted 1, but you can get after 20 minutes you can get a 2. Am I right? No, the 2 gene of interest. Okay, for that purpose only we are using this restriction endonuclease or restriction enzyme. Okay, remaining about the cloning vector we will see on another video soon. Okay, thank you guys. See you guys on another video. Thank you so much.